Molds growth found on breads and other bakery products are commonly Aspergillus and Penicillium species, but based on the observation of the bread for 7 consecutive days, it can be somehow concluded that the species is more to be a Penicillium species. The Penicillia of Penicillium species produce conidia from conidiospores that branch near the apex, forming a brush-like structure known as penicillus. The conidia of the Penicillia are colored, mostly in the shades of gray to blue-green, which is what the color of the mold grown on the bread. Furthermore, it has a fuzzy appearance and will produce mycotoxin, which will make it as a potentially dangerous mold. They can produce a number of mycotoxins, such as ochratoxins, penicillic acid, trichothecenes, fumonisins, and alpha-toxins. This image shows the simplest and most complex conidiophores produced by Penicillium species. To identify the specific groups of fungi, direct sequencing of PCR-amplified DNA using primers that are complementary to conserved sequence within the nuclear, mitochondrial ribosomal genes, and internal transcript spacer regions (ITS) were used. Among all of them, internal transcript spacer regions, as known as ITS, was favored for Penicillium species detection. Penicillium spinulosum, Penicillium roqueforti, Penicillium carnum, Penicillium verrucosum, Penicillium viridectum, and Penicillium aurantiogrisum are common Penicillium species found to be involved in bread industry. What are the most common sources of contamination and factors that affect the growth of microorganisms? Molds are able to convert nutrients into cell material and mycelial biomass. For instance, they may convert the carbohydrates and lipids to alcohol, organic acids, and complex heterocyclic biochemical compounds through secondary metabolism. This process was aided by the enzymes excreted from mold biomass to break the substrate down into available nutrients. Bread acts as a very suitable platform for the growth of fungi as it is high in both carbohydrates and lipids. These secondary metabolites accumulate in the bread and thus causing off flavors and odors. Mold growth are also affected by a number of factors, including moisture content, water activity, relative humidity, and temperature. All these factors are interrelated.